Good morning and good afternoon, everybody, and thank you for attending our webinar today, the Biocom Trail, a challenging hike towards animal-free testing. During the next 45 minutes, we will take you on a path through the history and future of animal testing, and we will highlight the milestones that we conquered through that path. As you will see, it has been a long and challenging hike, and we still have a difficult road ahead. But fear not, because today we have our true, very experienced guide to help you along this trial. I will leave Audrey, my US colleague, to present herself. So Audrey.
Hi, Peter. I'm excited for our hike today. I've got my pack and I'm ready to go. I've worked at Nelson Labs for over 25 years and spent about 20 years of that doing biocompatibility, doing the testing, and then also working in consulting to help write documents to support submissions.
And I am Peter Cornelis. I work at our Leuven facility, so in Belgium. I've been with the company for, well, almost 20 years, working in biocompatibility and microbiology, and currently I'm responsible for the investigation or the R&D that we conduct at Nelson Labs in Leuven for biocompatibility testing and in vitro testing. So to give you a few details on our hike today, we are talking about biological evaluation of medical devices. So every medical device that comes into contact with a human body poses a risk to that body. And it's an obligation for manufacturers to evaluate those risks and, if necessary, mitigate the risk that these medical devices poses to the body. All these risks, they are described in ISO 10993, and ISO 10993 is a very extensive guidance on all the risks, on all the possible risks that medical devices have on the human body. And ISO 10993 is a little older than 30 years today, and initially the way the testing was conducted, it could cost up to 100 animals that were required for all the tests that you had to do to release your medical device. Of course, that's not the way we like doing science at this moment. So we still want safe medical devices, and of course we would like to limit the animal suffering as much as possible. Certainly, if you know that over 95 or even 98% of all animal tests, well, they return a negative result. So it's a pity that we should inflict so much suffering on animals if the most of the part of the tests actually return a negative result. But also for the science part of the test, an animal test has several limitations, such as reproducibility. Of course, an animal is a biological organism. They do not always respond in the same way. So one species can, or one individual animal can react in a different way to an assay. So there is always the question about reproducibility. Also, most animal tests, or all animal tests, are qualitative tests, and of course, it's not always clear what the relation to humans is. At this moment, we know that several animals that are used today, they react differently from on the stimuli that we gave them, differently in relation to humans. So it's not always ideal testing on animals. This is our trail map. This is the very beginning of everything which has to do with in vitro testing. So in 1959, William Russell and Rex Birch, they published the principle of human experimental technique. And in that publication, there is the principle of 3 R's for animal experimentation. Although in the very early days of experimentation, there's always been controversy about the use of animals in testing. But even though there was very little regulation and very little harmonization. And Russell and Birch were the first to write down or to take initiatives to replace, reduce, or refine the test methods that were used at that moment. And replacing means that if there is an alternative test that does not require lab animals, then we should encourage to use them. And reduce animals means that if you have to use animal tests, we should try to limit the amount of animals that are required by optimizing our test methods. And then refinement also, if you conduct proper investigation, if you do your research well, if you refine your methods, then the number of animals should be limited, because we should try to obtain as much information as we can from one single assay and use as less animals as required. And this publication is actually one of the most important publications for animal-free test methods through the years. So during the presentation or during the webinar, we will first focus on what we already did, what we already achieved. Then secondly, how we test up to this moment. And then finally, what we still have to do to reach our ultimate goal, which is animal-free testing. So the regulatory trail, I want to show you this slide because it's a timeline of the most important publications from the last 30 to 40 years. And the reason I want to show you this is that if you look at the dates on the summits of the mountains, you will see that from the first legislation, which was published in 1986, well, in the early days, the first 10, 20 years after that publication, nothing really changed. It is not until 2010 that things rapidly started to change. And you can see that for the last 15 years or so, every 2 or 3 years, an important publication appeared. So the rate of the initiatives is really increasing. From the regulatory point of view, as I just explained, the EU directive from 1986 is the first real directive, the first legislation that was ever published, which handled animal testing. So before 1986, there wasn't any regulation, there wasn't any legislation on how animal experiments should be conducted. And the directive from 86 is a very extended one. It handles animal housing up to the proper conduct of studies, and it already encourages to limit the use of animal tests. But still, certainly for medical devices, this didn't change a whole lot. It was not up to the new update of this legislation in 2010 that things really started to change. And in 2010, two important updates were made to this directive. First of all, it anchored the principle of the 3 R's in the directive. And secondly, it described the establishment of an it established the Union Reference Laboratory for the Validation of Alternative Methods, which is now known as ECVAM. And only 2 years later, also the 3 R's were incorporated into the EMA. So if we look at ISO 10993, as I said, the first edition of ISO 10993 is from 1992. And as I explained in the first slide in the introduction, this is a guidance that contains over 23 paragraphs or 23 chapters, which all describe the different endpoints or the different risks that a medical device can have on a human body. And if you see at this table, this is a very important table that summarizes all these risks. And depending on how long and where a medical device comes into contact with a human body, you can see that the risks increase and that you should check more risks. And this is the table in this slide is actually from the update from ISO 10993-1 in 2018, which was a really important update, which Audrey will tell you in a while why this was so important. So Audrey, now up to you.
Well, thank you, Peter, for that introduction on where we have been. And now we want to talk about the trails we have recently forged, our current alternate trails. And so that's what I'm going to dive into next. First, I want to show you, I'm getting ahead of myself here. First, I want to just lay out our trail map that we're going to use. So this is just a simplified layout of the table that Peter showed in the previous slide. And this marks our biological endpoints that we need to evaluate when we have medical devices. And of course, the selection of these endpoints is based on the contact of the device, like how it contacts the body, and duration of contact. So we're not going to focus on that as much as we will how we can address these different endpoints. So I want to talk about a trail that we have developed for these long-term toxicities. So when we talk about long-term toxicities, we're really talking about subacute, subchronic, chronic toxicity, genotoxicity, carcinogenicity, and reproductive and developmental tox. Some things like acute systemic tox, those are not long-term, but you'll see as I go through this first part why those are lumped into this discussion. So you can see from our trail marker that we have two options to address these endpoints. So two trails we can take. We can do the in vivo, in vitro methods, and some of these do have some in vitro options, or we can do chemistry. So let's first lay out the in vivo options, which we have done for quite a while now. So you can see here, acute systemic toxicity. That's our quick one. So, doing that for an animal test, it's not a lot of animals, it's not very long. But then when you look at the other tests, the amount of animals increases significantly, and so does the time. And so this is something we want to take into consideration and think, is it really what we want to do is do the animal testing? So let's look at our alternate trail, which is the chemistry testing. And you can see when we look at that trail, we really don't have the need to use any animals, and we only have to manage one time point, which I think from a logistical standpoint, setting up these tests is also a nice benefit that is worth mentioning. So, this chemistry becomes a fast track. It's like riding a gondola right to the top of the mountain. Sometimes we feel like that's cheating, but in this case, it really is the smart thing to do. Even though it is easy, or it is fast, I just want to stress that it's not necessarily easy. So the thing here is we want to make sure that we design the studies correctly. So I'm going to give an overview of a few considerations that we look at when we design these studies, but I'm not going to go into a lot of details because that's too much for our presentation today. But the most important thing that we need to remember is that even though it's a fast track, it takes expertise to design these studies correctly. So these are the standards that we use to assess medical devices for chemistry testing. 10993-1 outlines options for a risk-based approach. So this standard gives us the guidance to do this fast track. So it's kind of like another trail map. -18 provides guidance on the chemistry testing for medical devices, and -17 provides guidance on establishing limits for the compounds that we detect in the chemistry testing. When we say chemistry testing for medical devices, we do mean extractable leachables testing. It's important to understand what are extractables and what are leachables. For the initial chemistry testing on medical devices, we typically focus on extractables. So these are compounds that can be extracted under exaggerated lab conditions. And when we do those exaggerated lab conditions, we do things like using multiple solvents, long extraction times, and high temperatures. This is to assess a possible worst-case exposure condition for a patient on whom the device is used. If the device cannot withstand the exaggerated conditions, then we will justify using simulated use conditions. But we have to try exaggerated first. So there are things that we consider when we design the study. So again, this is fast track, but we have to consider these elements. First, we always consider the medical device. What are the materials used? How does it contact the patient? How long does it contact the patient? Making sure while we're trying to do these exaggerated extraction conditions, that we design them correctly. So when we take those considerations of the medical device into play, we then design the extraction solvents. Typically, we use 3 solvents: a polar, midpolar, and apolar solvent. Then we look at time and temperature. Typically, we do like to use an exhaustive extraction, which has to be determined and then continued for all of the different solvents. Again, that's where we use that exaggerated condition. So if the sample or product can't hold up to that, then we can step that back to a more clinical relevant extraction. And then we look at an extraction ratio. Now this is set forth in 10993-18, and it's done using the analytical evaluation threshold. So the chemistry group looks at several factors in their equipment. So this is specific to each individual lab and each individual piece of equipment to understand, the appropriate sensitivity of the extraction. Then we take those solvents that are produced from those conditions and we analyze those extracts on our equipment. So one thing that's important to remember is that we are doing screening tests because medical devices are complex mixtures made of several different materials, different adhesives, different things in the manufacturing process. So the only thing that's targeted is what we do for metals typically. And so we are going to screen first for different volatility of compounds. And when we do that, we obtain data. Just a sec, I'm getting ahead of myself. We obtain some data that lets us know, okay, we have so much of each of these compounds. But in order to translate that to patient safety, we need a board-certified toxicologist to help us do that work. So that's where, when we do this fast track, it has to be paired with a toxicological risk assessment. So I have just a quick slide that shows us about toxicology. It's so much more complex than this, but this gives us an understanding that really the toxicologist is looking at the amount of compound that was detected from the device and comparing it to published threshold levels. So that's what all these acronyms are here is, where do we know we've seen adverse responses from this compound and at what dose has that happened? And then we see, okay, is the amount that came off the medical device close to that dose or is it far away? And then we kind of make a ratio and look at that and hope that we're very far from the dose where we've seen adverse responses. So this is a way that we're trying to use already published and known information about compounds. So that's what comes in our toxicological risk assessment. So let's just take a minute and look at our trail map. So I've identified where we have had these tests before, be animal tests. You can see that genotoxicity is not currently an animal test. It's already in vitro, but to save time and money and to make sure we're being as smart as possible for the testing setup, it can be combined with this. So you kind of get more scenery in your hike here is what we would say. Okay, so now, based on what we've talked about, our fast track here, we can change our map to show that now we've got a fast track option here for these endpoints. So let's just take a minute and appreciate the scenery of where we are. Look at our hike. So we still have some other tests that are using animal tests, or using animals for the endpoint. So I want to focus next on these three, we call them the big three: cytotoxicity, sensitization, and irritation. So these are our big three peaks that we need to climb next. Well, really, we've already done one, because cytotoxicity has always been an in vitro assay. But next, we need to focus on irritation. So there's some definitions on here just to help you guys understand the differences here. One thing that's important to remember is these are not predictive tests of each other. So if you fail cytotoxicity, that doesn't mean you'll fail irritation or sensitization. These are individual responses that we are looking at that produce results from different pathways in our body. So first, we're going to talk about irritation. And like Peter mentioned, there's been some progress on this one, and it's fairly important because it's taken a decade of work in the industry for us to pull this together. So this new standard, which is 10993-23, it's a result of over a decade of work in the industry and getting it published and making sure we demonstrate the safety and predictability of this test method. So again, just a definition for skin irritation. It's defined as the production of reversible damage of the skin following the application of a test substance. Now this says for 4 hours, that's in our actual assay. The key word here is that it's a reversible damage. So once you remove the irritant, the location of damage will repair. And we do this test in vitro using lab-grown tissue. So we're showing the lab-grown tissue here on the right, and then we have a tissue stained sample from an actual human tissue. So you can see what we want to focus on. You know, there's different shape between the two tissues, but what is the most important is that we have the same differentiation of cell layers, because each layer produces its own barrier properties that protects our skin and below. And we want to be able to mimic that in the lab. So you can see we call this a reconstructed human epidermis. Again, it's just tissues grown on a culture or on a filter. So this test, just to give you some parameters, it uses 15 tissues, and I'll show you. I don't know that we'll see those tissues today, but they're just very small tissues on a filter, and we want to do this in replicates. So it's nice that we can do this just on tissues on a filter. The data is quantitative because we are reading a color change, and we read that on a spectrophotometer. And the sample requirements are a little bit, you need the same number of samples, but they don't have to be as large for the in vitro option. And the turnaround time right now is still 4 to 5 weeks, which is similar to the in vivo tests. And the big reason for that is it takes 3 weeks to actually grow the tissues. And until we get the test more routinely, we can cut that down. But right now, until it's, you know, as widely as accepted as we'd like it to be, we're still sitting at that 4 to 5 weeks. So when we run this test, this is an example of the results that we look at. And the important thing on the chart or on the graph that you see is that we have a red line at 50%. And that's the cutoff for positive or negative. So in our table 1 shown here, this is for the GHS classification, so Global Harmonization System classification for irritants. So this test can predict a non-irritant or an irritant. And so that's, we just skipped the class 3 irritants, which are mild irritants here. But still giving us a lot of good data and information for medical devices, and a great opportunity to eliminate animal testing. So let's talk for just a minute about the acceptance of this testing. So right now we have the in vitro and in vivo models are both in ISO 10993-23. We can see that the US FDA has not gotten on board with the trail. I would say they're about halfway there. We keep hearing that we'll hear approval any day now. I feel like that's been a promise for a long time. But we are, as a working group and as a community, in conversation with the ISO or with FDA frequently to address any concerns they may have in the method. But you can see it is accepted widely outside of the US. So we've got great support internationally for this method. And so there's still an opportunity to use this cautiously, you know, with just some thought about where you're sending your product. Okay, so you can see that we have climbed 2 of our 3 peaks. And so now I'm going to turn this over to Peter so he can tell you about our 3rd peak out of these.
Thanks, Audrey. Well, sensitization, and as I told you in the early introduction, that's a little bit more complex and even more difficult than irritation. And as Audrey told you, this is the last peak of the big 3. And because we call this the big 3 because we need this for every device. Those 3 endpoints need to be assessed. And even not only the fact that they need to be assessed for each medical device is to be taken into account, but also because the predictive value of chemical tests is not so good for these 3 tests. So we really need them to be done. And currently, skin sensitization is tested by either the guinea pig maximization test, the local lymph node assay, and the Bueller assay. And as you can see, it takes quite a substantial amount of animals to do this. And it also takes a long time. So there are very expensive tests. They take a lot of animals, and they take a long time to complete. And as I said, it's actually a complex process, skin sensitization. I would ask you not to focus too much on every detail in this slide. The only thing that I want you to pick up from this is that, well, it's complex. And the reason why it's so much more complex than, for instance, the in vitro irritation test is that skin sensitization takes part in several areas of our body, and it uses a lot of different cell types are involved. And just to give you a few details, sensitization or an allergic response is divided into 2 events. You have sensitization and elicitation. And to explain this very easily, well, sensitization is an untrained response to a threat to the body. And you can compare this by shooting a cannon at a swarm of mosquitoes. It will take you a lot of time, it will take you a lot of effort, and probably you will not be able to get rid of all the mosquitoes. But your body learns from these events. And during the elicitation phase, well, we have our trained Marines or Navy SEALs that can attack very efficiently, very rapidly, and the likelihood of a success is much higher from our trained troops. So that's the elicitation phase, and that's we call that during what you get during elicitation, we call that the adverse outcome. So that's really the allergic response. And in that adverse outcome pathway, we identified a number of key events. You have key event 1, key event 2, key event 3, and key event 4. And these key events, they occur all in a different area of the body. So key event 1 is really at the top of on the surface of the skin. Key event 2 is a little bit more profound. Key event 3, even more profound in the skin. And key event 4 takes place after a transport through the bloodstream, and it takes place in the lymph nodes. And for these key events, certainly for the first 3, there are already 3 validated regulated tests available. but these are validated for pure chemicals, and that's really important to understand. So we're not starting from scratch. We already have validated assays, but as I said, they are validated only for pure chemicals. And of course, we're working with medical devices, and they are much more complex than these simple chemicals. So important from this slide to understand is that since the complete process of skin sensitization takes place in so many areas and implements so much different cell types, you cannot replace the in vivo skin sensitization by a single assay. So you really need a combination of several assays. And as I just showed you, you can find regulated tests that cover these key events. So for the 3 first key events, there are already validated assays available, and they are described in OECD 442. And probably, well, most likely, we've seen that if you cover at least 2 of those 3 key events or 2 of those 4 key events, you have covered most risks of skin sensitization. So we focus at Nelson Labs mainly on key event 2 and key event 3. And the last 4 or 5 years, we've conducted research mainly at the keratinosense method and the USense method. So again, very important to understand. These tests are currently only validated for pure chemicals and not for more complex extracts from medical devices. Because to test medical devices, you need to prepare extracts. You need to prepare extracts in an apolar medium. You need to prepare extracts in a polar medium, and of course, it's never a single chemical that leaches out of the medical devices. So you have complex mixtures, and we are not sure that these methods will react the same as they do on a pure chemical. ISO 10993-1 was updated in 2021, and from that time on, it recognizes the adverse outcome pathway with these 4 key events as I just described it. And as I said, they also mention that these in vitro methods, they have not been tested for chemical mixtures or not been validated for chemical mixtures. And we need to demonstrate if they are applicable for polar and apolar extracts. And the problem with a lot of in vitro methods is that they have to be conducted in an aqueous medium. An aqueous extract, or the aqueous test medium is not always miscible or compatible with certainly apolar extracts. And a lot of these methods, they can cause difficulties if you want to test apolar extracts. And to make things or life even more difficult to us, well, most sensitizers, they do not dissolve well in polar media. So we really need to get this apolar thing going. And the way we are conducting our research currently at Nelson Labs is this is just an example of a chemical that we tested in the keratinosense and the USense method. So what we do is we prepare dummy extracts of medical devices, and in those dummy extracts, so it should contain quite a substantial amount of chemicals that leach it out of this of the medical device, and we add intentionally known sensitizers. And at this moment, we have tested 20 different sensitizers in these extracts, and then we will try to see with the methods that we have. We make some adaptations or updates to the method so we can use them with apolar and polar media. And so we will try to find out at which concentration we can still find these or still can detect these sensitizers in our extracts. And that we compare to the data that we have from the in vivo assays. So what you can see here is this is the concentration response that we get from that particular sensitizer. So you can see that we have a concentration-related response. Line B is everything which is above this line is considered as being a sensitizer. So this is the threshold. And this line C is the sensitizing concentration for the animal test. So everything which, well, what you need to prove when you're assessing a method is that it's at least as good as the current method. And you can see, certainly for this chemical, that the in vitro assay is actually much more sensitive than the in vivo assay. And that's not a strange thing. That's something that we notice quite often. These are preliminary conclusions from the 20 chemicals that we have tested so far. We have had a lot of success with apolar solvents. So what is actually mentioned in the last ISO 10993-23 is that they fear, well, that's what they did, but that's what they write down, is that they fear that these methods might not always be compatible with apolar solvents. And we've noticed that for most chemicals we've tested, but I should emphasize it's most chemicals, and we have not have success with 100% of all chemicals that we've tested so far. But we've seen we had a lot of success with apolar solvents. And we notice little or no interference from the extraction mixture. So the fact that we are not testing the pure chemical, but a complex mixture, it does not seem to cause a lot of difficulties for these methods. And for most chemicals, it seems that these methods are much more sensitive than animal tests. So we are on a good track. But we still have we still have work to do. So sensitization, we're getting there. Let's say we're halfway there. And in 2023, important to mention is that an expert committee has prepared a draft guidance for validating these alternative methods. And probably early 2023, this will be published, and this will add the laboratories worldwide working on this to harmonize the approach. And we certainly well, as Audrey said, it took 10 years to get the in vitro irritation test validated. We're hoping that this will be quicker because we've learned so much from our experience with the in vitro irritation tests. Over again to you, Audrey.
Thank you, Peter. So now we can see that Peter and I have led you through some successful hikes. We've conquered a few peaks, but we definitely still have some peaks to conquer, like with the sensitization. So I want to take a minute and go back to our trail guide and look at, where we are and where else we need to focus. So we can see here where we've put in our fast track approaches. We talked about irritation being in vitro now with that option, and then sensitization we mentioned is halfway there, and that's a heavy focus for us, again, because of that big three. It will make a significant impact on the animal usage in this industry. So where are we looking after sensitization? So I've put a few of those in orange, which is the material-mediated pyrogenicity test. So there is a method that's already developed for pharmaceutical products. It's the monocyte activation test, and that's not been modified or tested for devices yet. So that's another direction we could go. And then, of course, hemocompatibility. There are some endpoints we look at within that realm that are in vitro, and we do have a blood loop method, but there's still some things that need to be addressed in that method to make it compatible for medical devices on a broader scale. So just to give you an idea of where we're looking next, implantation effects, I don't see that on the horizon. That may be one that we're not able to get rid of, just to be more protective of our patient populations. But you can see where we're looking next. So one thing I want to point out is, when we look at how we assess these medical devices, I want to say that it's helpful to have trail guides like Peter and I, so some consulting services. This allows the opportunity for some teamwork where we can take into consideration what we know about the device and these alternate trails to hopefully and potentially reduce the amount of animal testing. So that's one thing I think is valuable to keep in consideration as you're looking at your approach to assess your medical device. So we are, you know, very close. We feel successful about our hike so far. And as an industry, we are aligned to reduce animal testing. So I just wanted to put up contact information for myself and Peter. And Peter, do you have any comments about our hike today that you want to leave?
Well, it's been, as I said, we take pride in what we do. We take pride or we are try to be the forerunner in getting away from animal testing, and it took us a lot of effort, but we're sure we're getting there.
Excellent. Yeah, Peter and I are definitely forging these trails with our teams. There's so many people at Nelson Labs that are helping do this. So we're excited to talk about this. If you have any questions, please feel free to reach out via our emails. And then I just wanted to leave everyone with some options today, just to identify other partners. You know, these other companies that we work with, and highlight our other services and capabilities, including Sterigenics and Nordion, working together to safeguard global health. And then my last invitation here is that if your appetite for information on biocompatibility and chemistry testing is not satisfied, we want to invite you to go to our Sotera Health Academy, where we have established learning paths with free content available for you and your team members. So thank you so much for joining us today.